دكتورة مروة حميد الخفاجي، دكتورة في الأحياء المجهرية، قسم علوم الحياة، كلية العلوم، جامعة بغداد. مقرر التسمم الغذائي لطلبة الدكتوراه، فرع الأحياء المجهرية. محاضرتنا حول التسممات الغذائية البكتيرية Bacterial Food Poisonings. We know bacterial food poisoning classified into three categories: bacterial food intoxications, bacterial food infections, and toxico-infections. This lecture about bacterial food intoxications. The first sample or the first example is staphylococcal food poisoning, SFP. The first description of food poisoning caused by staphylococci is thought to be that of Vaughan and Sternberg, who investigated a large outbreak of illness in Michigan believed to be caused by cheese contaminated with staphylococci. Clear association of the organisms with foodborne illness had to wait until Barber at 1914 demonstrated that staphylococci were able to cause poisoning by consuming milk from a cow with staphylococcal mastitis. In 1930, Dak showed that staphylococcal food poisoning was caused by a filtrable enterotoxin. As a relatively mild, short-lived type of illness, staphylococcal food poisoning is perhaps more likely to be underreported than others. Most reported cases are associated with outbreaks. Only a few sporadic cases are detected of staphylococcal food poisoning. It is an intoxication caused by the ingestion of an enterotoxin preformed in the food. During the growth of certain toxigenic strains of staphylococci, mostly Staphylococcus aureus, the toxin is termed as enterotoxin because it causes gastroenteritis, an inflammation of the lining of intestinal tract. Staphylococcus aureus is a gram-positive cocci, catalase positive, oxidase negative, facultative anaerobic. Most enterotoxin-producing staphylococci clusters or cultures are coagulase positive cubes, produce thermostable nuclease. However, not all cubes are necessarily enterotoxigenic. Some of the toxigenic cocci are very salt tolerant, 10 to 20% of sodium chloride, and are fairly tolerant to dissolved sugars as 50 to 60% of sucrose. So these are the characteristics of the strain toxigenic or enterotoxigenic strain of staphylococci. This simple figure shows the staphylococcal food poisoning and the predisposing circumstances before the intoxication occur, where the chef must prepare food for 50 persons, so he must begin as early as 9 a.m. Preparing different types of stuffs of food containing eggs, milk, dough, cheese, meat, chicken maybe, vegetables, and the lunch may be consumed at 3 p.m. At 6 p.m. consumers get sick because of staphylococcal food poisoning. Rapid onset of vomiting after ingestion of reformed enterotoxin on bakery products, milk, cheese, or meat. This is a simplified figure of an enterotoxin production of food and causing staphylococcal food poisoning. Incidence in food. According to the Center for Disease Control Prevention, CDC, Staphylococcus aureus is a common bacterium found in the nose and on the skin of about 25% of healthy people and animals. So contamination by food handlers is also probably frequent occurrence in the view of high rate of human carriage as 25%. Since large numbers, typically over 106 per gram, are required for the production of enough toxin to cause illness, contamination is necessary but not alone sufficient to an outbreak to occur. So it is expected to exist, at least in low numbers, in any or all food products that of animal origin, or in those that are handled directly by human, unless heat processing steps are applied to affect their destruction. Milk and milk products including pasteurized milk, yogurt, chocolate milk, fermented milk, cream-filled pastries, poultry, fish, shellfish, meat and meat products, non-meat salads, egg products, vegetables, and cereal products have been involved. Symptoms of staphylococcal food poisoning include diarrhea, vomiting, abdominal pain. Rapid onset of vomiting is the predominant symptom. They are usually not life-threatening symptoms. Incubation period may be as little as 30 minutes or maybe 6 hours, but it is a short incubation period for staphylococcal food poisoning, 30 minutes to 6 hours to symptoms to be manifest. Most cases of staphylococcal food poisoning not require treatment because disease will pass on its own. And for that reason it is underestimated. Most people get over the food poisoning in about 2 days, according to the US Food and Drug Administration, FDA. Staphylococcal food poisoning related deaths are very rare. People at risk are increased risk may be complicated among elderly, small children, pregnant women, people with weakened immune system, what we call immunocompromised patients. The minimum quantity of an enterotoxin, staphylococcal enterotoxin, needed to cause illness in human is about 20 nanogram. The figure shows mechanism of staphylococcal enterotoxin, how it induces emesis or vomiting. Firstly, enterotoxin directly affects intestinal epithelium, then stimulation of the vagus nerve, then transmit signal to the vomiting center in the brain to produce a large quantities of vomiting. Detection: colonial appearance on either mannitol salt agar, where it is mannitol fermenters, or on Baird Parker agar, and this is the powerful detection mean by Baird Parker agar gives presumptive identification of Staphylococcus aureus, which is often confirmed them by production coagulase, thermonuclease, thermostable nuclease. On Baird Parker agar, Staphylococcus aureus appeared as a black colony. This figure is from my work. ELISA technique, which detects 0.1 to 1 nanogram of toxin per gram of food, and reverse passive latex agglutination test with sensitivity of 0.5 nanogram per mol. The second type of bacterial food intoxications is botulism. Because of its severity and distinctive symptoms, botulism is the form of bacterial food poisoning for which we have the earliest reliable reports since 1793, where 6 people died after eating sausage. Kerner noted that several important features, including facts that heating was an essential precondition for the development of toxicity, enhanced toxicity, heating the food. In sausage, and small sausages that containing air pockets were less likely become toxic. This was the first report about botulism. It was identified with ichthyosm, a paralytic illness associated with consumption of raw salted fish known in Russia in 1880, and in 1904 an outbreak of botulism in Germany was caused by canned white beans. 4 botulism syndromes are recognized. The first one only related to food. Foodborne botulism caused by ingestion of food contaminated with botulinum toxin. Wound botulism caused by Clostridium botulinum colonization of the wounds and in situ toxin production. Infant botulism caused by intestinal colonization and toxin production. Adult intestinal toxemia botulism, an even rarer form of intestinal colonization and toxin production in adults. Inhalational botulism could result from aerosolization of botulinum toxin, and iatrogenic botulism can result from injection of toxin. This what may occur when using botulinum toxin Botox in plastic surgery, iatrogenic botulism. Only the first type, as I mentioned, is a food involved type. Botulism is an intoxication caused by ingestion of food containing botulinum toxin, which is a neurotoxin, unlike enterotoxins of staph, which act locally in the gut. They affect primarily the cholinergic nerve of the peripheral nervous system. Botulinum toxin produced by Clostridium botulinum gram-positive anaerobic spore former rods with oval or cylindrical terminal spores. Mode of action of the toxin. The toxin ingested with food and surviving inactivation in the stomach, absorbed in the upper part of the small intestine, reach the bloodstream via lymphatics. Mode of action. The toxin ingested with food and surviving inactivation is absorbed in the upper part of the small intestine, survive inactivation in the stomach. It reach a bloodstream via lymphatics, bind to the nerve ending at the nerve muscle junction, blocking release of the acetylcholine responsible for transmission of stimuli, thus producing a flaccid paralysis. Incubation period. Initial symptoms of botulism occur from 8 hours to 8 days, most commonly 12 hours to 48 hours after consumption of the toxin containing food. Clinical symptoms include vomiting, constipation, urine retention, double visions, difficulty to swallowing, what we call it dysphagia, dry mouth and difficulty to speak, dysphonia. The patient remain conscious until, in fatal cases, shortly before the end, when the progressive weakness results in the respiratory or heart muscle failure. This usually occurs in 1 to 7 days after the onset of symptoms, depending on the dose and on the circumstances related to the patient. From 1 to 7 days. Surviving patient may take as long as 8 months to recover fully. Botulinum toxin is a protein of high molecular weight, 150 kilodalton, powerful, so powerful that only tiny amount, 1 to 2 microgram, is sufficient to cause death. It is absorbed mostly in the small intestine, paralyzing voluntary muscles of the body. The toxin relatively stable, being inactivated only at 80 centigrade for 10 minutes. The heat treatment necessary to destroy this toxin depend on the type of organism, the producing organism, the strain of Clostridium botulinum that's producing this toxin, and the medium, and in this case the food. Acidic food more resistant than other foods. For example, the medium in which the toxin is heated. So 80 centigrade for 10 minutes, depending on the organism producing this toxin and the medium in which is heated. Thank you for listening.